Good afternoon, everybody. My talk will be in English. Sorry for that. So I hope you can follow. If at some point you want to discuss more some details of the slide, I will be available after, of course. So I am the R&D director of the R&D in Grifols company. So we are specialized in immunohematology. So this talk probably will be different from what you have seen before, because you have discussed a lot on clinical aspects and diseases, and this talk will present to you how we develop a new product. So I will take you through the journey of development of a new product in the company. Okay. So this you know perfectly. I think it's your perfect expertise. About multiple myeloma, which is a blood cancer that develops in plasma cells in the bone marrow. So in multiple myeloma, plasma cells are transformed into cancerous cells, and these cells grow out of control and they crowd out the normal cells that help fight infection. So reduction of blood cells can cause anemia, excessive bleeding, and decrease ability to fight infection. CD38 is widely expressed on myeloma cells, and it is expressed only at a low level on other...
Sorry for interrupting. Can you talk to the microphone?
Sorry?
Talk to microphone.
Okay. Is it better? Yeah? Okay, sorry. So I was saying CD38 is widely expressed on myeloma cells, and it is only expressed at very low level on other cells in bone marrow, and it makes it easier to recover after therapy for these other cells. So when Darzalex binds to CD38, it causes cell death in multiple ways. So it kills myeloma cells directly. It also recruits immune system cells called macrophage. So macrophage bind to the Darzalex CD38 complex, and then this macrophage can destroy myeloma cells. There are many ways. Probably I will not go over all of them because we are ahead schedule, and I will move to the next slide. Okay. So, history of drug development for multiple myeloma. We have first the European Medicine Agency, the EMA, who is responsible for the scientific evaluation, supervision, and safety monitoring of medicine. So Darzalex received from EMA first conditional marketing authorization in 2026. And then it was switched to a full marketing authorization in 2017. For FDA, Food and Drug Administration in the United States. So in 2016, the FDA granted as a prior therapy the daratumumab, the Darzalex, in combination with other drugs for the treatment of patients with multiple myeloma. And it has had a priority review, and then it was granted current approval in 2017. So in Grifols, in the R&D, what do we do when we start a new project? So first we gather inputs from the literature to understand everything around the drugs, around the use, around clinical therapies. So I put here, for example, 2 reports from the literature in the field. And then we noticed that despite we knew that daratumumab can cause interference in pre-transfusion immunohematology testing, the laboratory community was unprepared. So this drug was introduced in the clinics, and then we could see that it impacted, I will show after, pre-transfusion immunohematology. So we know that this interference in immunohematology testing is a particular problem in patients because these patients require RBC transfusions during the disease treatment. So the antigen CD38 is also expressed on the surface of red blood cell. So what's happening, is it here? So you have here a normal red blood cell that is expressing CD38, and the patient serum contains the daratumumab. So what is happening is daratumumab binds to CD38 on the red blood cell, and when you add the Coombs reagent, so the anti-IgG, to perform antibody screening or antibody identification, you get an agglutination, a false positive due to daratumumab. So you can see here, it is a routine testing where you can detect a specific profile with an alloantibody. But in the case of daratumumab, we can see an interference. Everything is positive, and you cannot distinguish any antibody in the plasma. You get a false positive with all the cells that you use for antibody screening or identification. So in the labs, the real problem is anti-CD38 contained in the plasma. It is created an interference in immunohematology testing, and then it is masking the potential presence of an alloantibody that we want to detect. Okay. So then labs started to develop some techniques to neutralize anti-CD38 in the plasma. So we have, for example, treatment based on RBC treatment. So for example, we have the RBCs that is used in the panel for screening and identification that we will treat with, for example, DTT or trypsin or mercaptoethanol, so any enzyme. So because of that, we will destroy CD38 on the surface of the red blood cell. Then another strategy consists in using Fab fragment. The Fab fragment will hide and cover CD38 on the surface of the RBCs. And then when you mix with the plasma, if you have, for example, here any therapy anti-CD38, it cannot attach to the antigen because it is already covered with Fab fragment. Okay? Then labs are using also antibody adsorption. They use cells to adsorb anti-CD38. It can also be used a recombinant soluble protein. It is the approach that Grifols used. And then some labs would use specific cells that do not express CD38 on the surface, for example, cord blood cell or in-loose cells. We can find in paper the main difficulties related to the use of these techniques. So mainly when you would use DTT, it can denature not only CD38, but also KEL antigen and other potentially clinically significant antigens, like Lutheran, Cromer, NOPS, and others. If you use in-loose cells or cord blood cells, they are rare and not routinely available reagent cells. Then these techniques requires training and competency of technologists. It is techniques that are time-consuming and cumbersome. It is not easy to run. And then the methods are not standardized. So Grifols' proposal was to design a recombinant CD38 to neutralize the interference. So we generated a recombinant soluble CD38 that we call S-CD38 with a high affinity epitope for anti-CD38, like Darzalex. So what we do indeed is we have this patient serum containing daratumumab. We add soluble CD38, we incubate, and then we block soluble CD38 can block the daratumumab present in the plasma. So after you have blocked the antibodies, you can run the testing as usual, antibody screening, antibody identification, or cross-matching. So you have again red blood cells that express CD38. You can mix with the patient serum, but this time the anti-CD38 is blocked. And because of that, when you add the Coombs reagent, you do not provoke anymore the false positive agglutination. Okay, so it has been done some work in, we have facilities in California, in the USA. It has been done some work to design really this soluble CD38 so that it has a very high affinity with the anti-CD38, the therapeutics. So you can see here, it is the design that we have chosen because it has a very good neutralization rate with daratumumab. And then we were checking on SPR, the binding kinetics of CD38 with daratumumab, and we could see and measure a very good affinity. Okay. Then we move to the project approval internally, so to start developing this reagent. So many departments are always involved in this project. So for example, we have marketing team who would help build market analysis, they will do forecast, business case, and they will do an analysis of competitors. So when this is done, we approve the project and marketing is preparing target product profiles. So in this document, they will state and claim everything we need to develop the product. In R&D, we have two sites that worked on the product. So the site I have been showing in the USA called GDS, Grifols Diagnostic Solutions. So this site has the expertise to product and develop recombinant protein. And another site in Switzerland, Median Grifols Diagnostic, it has immunohematology expertise. So both these two R&D sites were able to develop and offer proteins soluble to pre-treat plasma from DARZALEX patient and to neutralize anti-CD38. And then we have done also some intellectual property research, and we have patented this soluble CD38. Regulatory affairs help us defining regulatory requirement to have this product, for example, the CE mark, or to register this product in any country we are interested in. And of course, finance department helps us to budget, the cost of this development work. Okay, so we started with the feasibility activity. So GDS was responsible of developing the constructs, of producing the protein, of purifying the protein, and scale up the product. Because if we need a lot of product, it cannot be at lab scale. It needs to be scaled up in manufacturing area. And then Median Grifols Diagnostic in Switzerland was receiving the construct and could develop the final formulation and all the testing method. The idea here is to optimize the neutralization effect but avoid dilution of plasma, because of course if you put too much product in the plasma, you dilute plasma and you are at risk to lose detection of weak antibodies. So it's a balance between good neutralization effect but not too much product. Okay, then it was developed 5 different methods that we can see here. So for the gel card technique, we have different methods for antibody screening, identification, and cross-match. I will show later. We can do either the treatment of the plasma with the soluble CD38 inside the gel card or outside the gel card. And we have also developed a method to use the product in the glass tube, conventional tube technique. Okay. So this is just, we listed here the reagents that are needed to perform the technique. So for example, the red blood cells, the gel card, incubator, and all usual, reagents needed. Okay, so this is drawing to explain how we use the product. So we have here what we call the internal pretreatment. So we use the card as always. Inside the card we can put 4 microliters of Grifols CD38, and then we put 25 microliters of the plasma or serum of the patient who has received the therapy, for example. Then we incubate 15 minutes at 37 degrees so that the soluble CD38 can block anti-CD38 in the plasma. And then we can continue with the usual steps of an antibody screening. We add the reagent red blood cell, we incubate, we centrifuge, and we read the results. We can also use what we call an external pretreatment. This time we would mix the soluble CD38 and the plasma, not in the card, but in a glass tube. So we mix, we incubate, and then we do, as usual, the steps of antibody screening in the gel card. So we add red blood cells, 25 microliters of the treated plasma with the blocked anti-CD38. We incubate, we centrifuge, and we grade and record the results. So this is an illustration of what we can obtain. So for example, you can see here we have plasma of a patient that contains an anti-KEL antibody, and also he has received Darzalex therapy. So if we do not treat the plasma, you can see you have false positives for any red blood cell used. After we treat the plasma with soluble CD38, we remove interference due to Darzalex, but we can still detect the anti-KEL antigen on cell 1. So these are results with one card and with another type of card. So here the idea is to say that soluble CD38, the treatment, will get rid of the interference due to Darzalex and will reveal the presence or absence of irregular antibodies, because for sure we want to see irregular antibodies. Okay. So after feasibility step, we started the development of the finished product. So I listed here example of studies that we do internally. So it is, for example, we set up the manufacturing and quality control procedures. We validate. We need to validate suppliers. We manufacture what we call pilot lots. We check the stability of the product. And then we do any other studies needed. For example, we check that if we send the product abroad, it's okay. I mean, the product stands the stress of a shipping, Then we design the labeling and the packaging. We do a risk analysis, and we train the other departments, and we prepare all the documentation needed for regulatory registrations. So I have put here an example of studies that is required to be done. We can take an example, for example. We will check, for example, during all the studies, what we call reproducibility. This, I mean, is to check that if I perform the testing one time, if I repeat the testing, I get the same results, because it is important, right? So to do this reproducibility study, we will use 2 lots of Grifols soluble CD38, 2 different operators, and we will do 2 runs per day during 5 days. So in total, 20 replicate. And the 20 replicate need to give the same result so that we can be sure product is working same with any operator. Then we did clinical performances. So the clinical performances is we compare our product with another method. So in that case, we compared the soluble CD38 with DTT method. And in case of discrepancies, to understand which technique is saying the truth, we used the DARA-X. And from the results, we do the comparative table. And we calculate a positive agreement and a negative agreement. And the 2 methods need to have a very good sensitivity and specificity. We also added 500 samples. These 500 samples have to be tested with at least 10% of patient samples, because it's not always easy for us in the lab to supply real patient samples. So what we do is we use 10% of patient samples, and then we complement with what we call spiked plasma. Means we have plasma of donor or patient not receiving the drug, and we add the Darzalex that we supply from the pharmacy. So we simulate a patient that has received the therapy. Okay? And then on top of that, we did like mixes with main alloantibody specificities we want to detect. So what we do is we check that after the treatment with soluble CD38, we remove interference with daratumumab, but we can still detect these antibodies, which is important and which is the aim of the product. Okay? So then when every study has been completed, we move to CE mark of the product. So we submit what we call a technical file to the notified body in May 2022. So then the notified body tells us it is okay, you comply with IVDD, so you can start selling your product. So after we got IVDD, we wanted to understand if customers would be happy with the product, so we did what we call a pre-launch study. So for this study, we included 8 European centers, 1 in UK, 3 in France, 1 in Spain, 1 in Switzerland, and 2 in Italy. So we sent the product to these 8 centers, and we asked them, can you use the product and tell us if you are happy with the product or if you are not happy. So in total, 273 samples collected. So these sites could treat the plasma, as I have been showing, so with different volumes, 2, 4, and 6, 25 microliters of plasma, incubation, and then they would do antibody screening in gel technique. Sometimes when the concentration of Darzalex in the patient is very high, 2 microliters is not enough. So the user needs to move to 4 microliters. And if he still has interference, he can use 6 microliters, not more than 6, because if he is using a lot of volume, it will dilute antibodies. So 2, 4, and 6. So these are the results. So in total, 273 samples, 10 isatuximab samples, and 263 samples of patients treated with daratumumab. So you can see here the percentage of the neutralization with 2, 4, or 6 microliters of soluble CD38. So what we can see is for isatuximab, it was okay with 2 microliters and then 4. We can inhibit 100% of interference. For daratumumab, we inhibit around half with 2 microliters, around 25% with 4 microliters, and 20% need 6 microliters because of concentration of the therapeutics in the plasma. So we can see that for a patient that have a high concentration of drug in the plasma, we could not neutralize, only partially, so 1.5, okay? So soluble CD38 could neutralize daratumumab in 98.5% of the sample, and the isatuximab in 100% of the samples. So some sites did a comparison study with DTT because some sites were already using DTT. So you have here the results. So the DTT could completely neutralize the interference caused by anti-CD38 in 78.35% of the samples. Okay. So you can see here the detail in the site using DTT method. Then we checked antibody detection. So this is the table of antibody detection with soluble CD38 treatment. And this is the table with antibody detection after DTT treatment. So you can see here that when we used soluble CD38, we lost anti-Duffy A and anti-Duffy B detection. Okay? So in the IFU of the product, there is a limitation, and we say when you use soluble CD38, you are at risk to lose anti-Duffy. In the case of DTT, we can see that we missed more antibodies. 5 antibodies were missed. Anti-Big E, anti-Duffy A, anti-KEL, anti-D, and anti-KPA. We have then requested the 8 sites to fill a survey, right, a satisfaction. So they had to score out of 5 points if they are satisfied with the use of soluble CD38. So you can see they were quite happy with the reagent. It's easy to use. The shelf life is long, preparation is rapid. And then here they rated low, probably the anti-Duffy limitation, but still they say that it can be overcome by providing Duffy A and Duffy B matched unit, because if you cannot detect anti-Duffy A, you have to find a solution, a mitigation. And then they say that probably 8.4 out of 10 would recommend this product to colleagues. Okay. So as conclusion, the treatment of plasma allows the further use of different IH methods. It's easy to use, and it's quick compared to DTT. We pointed out that maybe the drawback is that it is requiring anti-Duffy A, and Duffy B matched units. It has a higher efficiency than DTT in neutralization of anti-CD38, both for daratumumab and disatuximab. And it is CE marked with IVDD, and then we did studies to get CE mark with the new regulation, IVDR, the directive. Okay. So we submitted the IVDR submission in 2024, and we did some improvements on the reagent. So for example, now it is possible to treat the plasma sample and to use automation. And we also added in the instruction for use the possibility to use the 6 microliter method. Yeah, so thank you very much. Thank you.